Hello everyone, and welcome to today's webinar, Make Your Research Count, Generate Reproducible Results with Recombinant Antibodies. I am Jennifer Woods, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more about our sponsor, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Dr. Urmi Roy, Global Product Manager for Flow Cytometry Reagents. Dr. Roy, you may now begin your presentation.
Hello everyone. Thank you for joining us today. We will be discussing how recombinant antibodies can enhance your research reproducibility. To those of you who are not familiar with Miltenyi Biotec, I have this short summary here. Miltenyi Biotec was founded in 1989, and over the last 30 years we have grown significantly, and our pioneering technologies have constantly been supporting research works, which you can see from more than 46,000 scientific citations. Our continuously growing product portfolio ranges from basic research to clinical applications and ensure true translation. For today's presentation, I have the following agenda. I will discuss about reproducibility crisis and pitfalls and why antibody quality and validation is important. Then I will introduce you to our recombinant antibodies and show how they can improve quality of research. I will highlight the importance of antibody validation for research and how here at Miltenyi Biotec we support you with that. And I will sum up with some final consideration and summary at the end. So to start with the first point. In the past years, there is a growing concern within the scientific community that many research results are not reproducible. In 2015, a study published in PLOS Biology showed around 50% of the preclinical research spent in the US are basically wasted due to irreproducible results. And they have categorized the contributors of such irreproducible data, and biological reagents were shown to be one of the biggest culprits among the others. When talking about antibodies and that they are considered as one of the key variables that attribute to the reproducibility crisis, I would like to show you some examples here. In 2008, in Human Protein Atlas project, found that less than 50% of the commercially available antibodies recognized their specific targets, which means the rest were actually unspecific. And in 2009, in Dr. David Ream's lab was unable to continue their cancer biomarker study due to antibody lot-specific irreproducibility. Followed by in 2012, Amgen tried to redo some previously published preclinical research studies and only could reproduce 6 out of 53 studies. So all these examples led to a very strong debate in the following years about the antibody-related research reproducibility and transparent reporting of their use. And later on, several reports came up where need for better technology were discussed among scientists, companies, as well as organizations like NIH, and recombinant antibodies were discussed to be used to tackle such a situation. So what are exactly the facts that affect the antibody performance in flow cytometry and results in a puzzled research outcome? Here I'm showing you several important factors that are including the unspecific binding of antibodies resulting in background noise, lot-specific variability, insufficient sensitivity, as well as poor characterization and lack of reagent information These are also very important factors that affect the performance. Using reagent without knowing how they will perform under specific condition as well as experimental protocol may also contribute to data variability. And finally, use of reagent from different sources means that you have to use different control, and without such proper control, you cannot ensure the performance of your data. So what can we do to overcome all these kind of factors that affect the reproducibility crisis and that are originated from antibodies? We believe there are three solutions. Use of advanced technology to create better reagent, proper validation of the reagents, and thorough documentation that can be followed by the fellow researchers. We at Miltenyi Biotec are trying our best to support you with reagents produced using the best technology. And together with the support from the scientists in the validation and documentation effort, we can battle with the reproducibility problem with the advanced technology. This is where I would like to introduce you to the recombinant antibody technology. At the moment, there are three prominent categories for antibody generation: polyclonal antibody, hybridoma-derived, and recombinantly derived monoclonal antibodies. And with this chart, I am showing you a very summarized way of differences among these three categories. So polyclonal antibodies are basically a mixture of heterogeneous antibodies that recognizes several epitopes, resulting in a stronger signal, which is obviously important for certain applications. These are purified from serum of immunized animals and offer very low lot-to-lot consistency. And therefore, it's not really a very good, suitable antibody for flow cytometry. On the other hand, monoclonal antibody derived from hybridoma are actually mixture of homogeneous antibodies that recognize only one epitope, thereby ensure higher specificity. These are purified from single clone of immortal hybridoma cell lines and also offers lot-to-lot consistency. Therefore, it's a good choice for flow cytometry application. Finally, the recombinant monoclonal antibody, they are genetically defined antibody. I will show you later a little bit more in detail. This means that they are defined on their DNA level and recognizes also only one epitope with their highest specificity. And genetically engineered antibody together with a defined expression production system ensure the highest lot-to-lot consistency and thereby also the best fitted antibody for flow cytometry application. So let's have a closer look at the manufacturing process of hybridoma versus recombinant antibodies. Hybridoma clones are generated from fusion protein from an antibody producing B cell and immortal myeloma cell. They are not pure because they contain light chain produced by myeloma cells, which are also B cells here. And you can see with the green, with this green light chain contaminant. And also, in case this kind of cell line are cultured over long term with many passages, this can lead to spontaneous mutation, and that results in antibody clone which is different than your expectation. Further contamination can also happen coming from the culture media, which are also basically from serum, and then led to some immunoglobulin impurity in your resulting antibody. Therefore, all this leads to a lot-to-lot variability as well as decrease in the purity of the product. Recombinant antibody, on the other hand, are genetically engineered. They have a defined DNA sequence which is inserted into mammalian cells that are programmed to express the product of this particular DNA sequence. Since they are not derived from B cell and also cultured in a defined condition, the contaminants that you have seen in the hybridoma clone scenario is not there for the recombinant antibodies. These allow higher batch consistency and purity. An additional benefit include the ability to engineer these antibodies with the highest flexibility. And this I will show you later in the slides that how Miltenyi Biotec took the advantage of this technology of recombinant antibody to generate high quality product. Now, since 2012, we are offering our recombinantly generated antibody that we call Re-Affinity antibody. And this antibody offers superior technology as well as high quality production. And in the next slide, I will shortly outline some of the key benefits that comes along with this technology. So when we're talking about the technology itself, and here you can see a model of our Re-Affinity antibody. Here we took the advantage of the genetic engineering to generate this antibody, which contained the antibody variable region, here depicted as orange. And this is derived from hybridoma clone, which has validated application for flow cytometry. So they are very specific. Then you have the backbone, which here in purple are deriving from the constant region of human IgG1. So all our antibody basically contains the same IgG1 backbone. Finally, we have used the Fc region of the backbone. We have done some mutation there to abolish their binding to Fc gamma receptor, and I will show you later what are the benefits with these kind of mutation. So first of all, as highlighted earlier, that recombinant antibody generation results in highest purity. Here is an example of mass spectrometry data where you can see clearly that recombinantly generated antibody shows highly pure antibody compared to your hybridoma clone, where you have this pink star that is showing some contaminating light chain. And in this way, we are really highest purity product. Now, when I told you about this mutation about the Fc region, here is some data where you can see that there are 5 different Fc gamma receptor which were tested, comparing our Re-Affinity antibody in light blue and the monoclonal hybridoma antibody in dark purple. And you can see that our hybrid, compared to hybridoma antibody, our Re-Affinity antibody showed very slight or significantly lower binding to this unspecific Fc gamma receptor. And now, why should you care about this kind of binding to Fc, all these Fc gamma receptors? And this I will show you with a customer provided data, thanks to our customer who has shared with us this data. And here you can see in the plot on the left-hand side, where you can see from hybridoma clone, which offers the which shows this unspecific binding marked with this red symbol. And you can see that it's not consistent in all hybridoma clones. So one hybridoma clone 1 did not show this binding, unspecific binding, compared to hybridoma clone 2, and without any SCR block. On the other hand, in the Re-Affinity clone, you don't see this background. So of course you can use the SCR block reagent, which are commonly used in the field, to get rid of this kind of unspecific binding. But this does not ensure the perfect scenario, because sometimes here in this case you see that you could not get rid of it completely. And this obviously includes unnecessary additional experimental variability that comes along with the FCR block reagent itself. So if possible, it's better to avoid such experimental condition. Finally, I showed you that our recombinant antibody contains universal human IgG1 backbone. And what is the benefit with that? Ideally, when you use hybridoma-derived monoclonal antibody, they probably were generated with many different background. For example, mouse IgM, IgG2, and so rat IgG1, or so on. This means for every individual isotype, you have to make a proper control for them. So this leads to a complex experimental planning and inventory management. Compared to that, in case of Re-Affinity antibody, due to the fact that we have a universal backbone, you only need, in such a case, you only need one isotype control, and that allows you to perform a straightforward experimental planning as well as inventory management. Now, in the next part, I would like to briefly touch about the high quality production of our final antibody product. And our production procedure follows a strict steps to ensure highest quality, and throughout the process, the quality is thoroughly monitored. And the relevant information we also provide to customer for specific regulated research that is very important. Here is a brief outline of our antibody purification process. Antibody and fluorochrome, both as the starting material, are generated with strict quality control. And upon the highly qualified starting material, we perform the coupling, and then we perform thorough purification step for the conjugate to get rid of unbound antibody as well as unbound fluorochrome to ensure highly pure, only conjugated antibodies in the final product. And we also perform functionality tests on primary samples to test the performance of the antibody, and these we maintain across all the lot. Here I must mention that this also is performed with the titration. What does this mean? That when you have performed such a lot specific experiment, this leads to product that will perform the same, exactly same in every antibody lot. And with this data set, you can see for CD56 conjugated to different fluorochrome, we have compared two different lots, and you can see their performance is exactly the same in this histogram. And as I mentioned, every lot is tested and titrated for optimal performance, and thereby we ensure the high lot-to-lot consistency, as well as reduce your effort to every time perform a titration experiment for every lot of antibody. As mentioned earlier, you can easily access also our additional documentation such as certificate of analysis for respective product lot after purchase, and you can easily download them directly from our COA tool in the website. So here, since I mentioned since 2012, we have started with this recombinant antibody portfolio, and over the years we made sure that we cover all the markers, and we are proud to say that we have the largest portfolio of recombinant antibody at the moment, which are optimized and validated for flow cytometry, which includes more than 1,000 clones. And also as a final product, our antibodies are conjugated to our highly bright biodyes family, and all together we have more than 16,000 fluorochrome conjugated products, which are the specificity ranges from different cross-reactivity from human to rodent as well as non-human primate. And as I mentioned, we cover a lot of different markers that are cell-specific and also different applications like immune checkpoint, cell signaling, cell death, and more markers are covered within our portfolio. Just here quickly to mention that we also are increasing our portfolio and testing them beyond flow cytometry, specifically focusing on their application in imaging as well as mass cytometry. Now, the next part of my talk, I would like to emphasize on antibody validation and our company effort on this topic. So several key opinion leaders in the antibody field has discussed and defined the standards which are required to achieve highest quality antibodies. This includes reproducibility, specificity, and sensitivity. And our aim is to use the available tools to validate all our antibodies and communicate this to our customer in a very transparent manner. So in the next slides, I will show you what are these steps that we are taking here at Miltenyi Biotec to achieve these validation standards. So I would start with the reproducibility, where in the field it's often discussed about the lot performance of the antibody. And here I won't go so much into detail because these arguments for us being as a company where we ensure the highest reproducibility is coming with the benefit of our Re-Affinity antibody technology. So starting from the recombinant antibody technology until the manufacturing, we can achieve the highest reproducibility level by lot-specific reagent with all the benefits that I already mentioned earlier. So you can definitely be sure that our antibody offer you the highest reproducibility standard in this regard. I would like to go more detail into the topic of specificity. This is a very important point because often there is a lot of research which cannot be reproduced because the antibody was not specific enough to the target antigen. And therefore, this is one of the mostly debated topic in the field. So what we are doing to ensure the antibody specificity of our Re-Affinity antibodies. So we do several experiments. During the individual product development, we perform suitable counter staining to verify that the antibody is actually binding to the target population of cell of interest. Moreover, we perform assays with advanced technologies such as CRISPR-Cas to use knockout of target protein, as well as we perform Epitope computation assay. This I will show you a little bit more detail later. Moreover, for specificity that require additional assays, there we also perform tests like siRNA knockdown. We perform tests like stimulated cells, overexpression of specific target protein, as well as we perform experiments like binding of purified antigen that are coated in latex bead. Now, as I said, that in the field, the really high quality specificity tests are recognized to be validated by assays such as the knockout of target protein, as well as testing their epitope recognition. So what are we doing here at Miltenyi Biotec to confirm that? So to test this antibody specificity, here our aim is that we generate cell lines where we knock out the exact protein of interest from the cell, and we compare the performance of the antibody to wild type versus knockout. And we also display this data in our website. This first data set that you can see here is coming from a microscope, and you can see that in a wild type cell line, the target antigen is present, so the antibody is showing very nicely staining here in red, and in a knockout cell, this is completely gone. This means that our antibody was highly specific for this target antigen. And we also demonstrate this kind of data using flow cytometry. For the red histogram here, you see the red, which is wild type, and then for knockout is the blue. So the benefit for you here is that you need less validation. You can trust on this antibody, and this also gives you confidence about the quality of the antibody. So the second test that we perform is the epitope computation assay. Our recombinant antibody clones have specific name with REA at the beginning. And we are aware that this might be difficult for you to recognize the binding of our REA clone compared to other known hybridoma clone. So we perform tests to monitor whether our antibody is specific to certain epitope recognized by other hybridoma clone or not, and thereby build your trust on our REA clone. And the method that we follow here is basically a blocking experiment. Here in the explanation chart, you can see that in a regular staining, of course, when the antibody is conjugated to a fluorochrome and you stain your cells, it shows you a 100% signal. In a computation assay, we use pure antibody of our Re-Affinity clone, and we stain the cells with this clone to to completely block the epitope. Now, what happens in this case? Then afterwards, we perform a staining with the fluorochrome conjugated hybridoma antibody to see if they can bind to antigen or not. In case the REA clone do not share the same epitope as the hybridoma clone, this will result in a successful staining in flow cytometry, here as you can see, with no overlap, meaning that the hybridoma clone was basically targeting another epitope. In case the hybridoma clone and the Re-Affinity clone share the same epitope, the pure antibody, the Re-Affinity antibody, will completely block the epitope. Thereby, the fluorochrome conjugated hybridoma clone will not be able to bind, and you will not see any signal. And in the website, we display this data with this kind of table. So in our product page where we have this kind of data, you will be able to see, compared to the existing hybridoma clone, how our Re-Affinity clone is performing in terms of their epitope recognition. What is the benefit here for you? You can easily switch from a clone that you were using before as a hybridoma, and you can find the matching Re-Affinity clone for your research. And you can trust on this because they are basically recognizing the same epitope. And you can also compare the epitope recognition of several clones at a glance. So this data actually is valuable for you also to see the other clone performance. Now the final part here is the sensitivity of the antibody. And this basically means to check the performance of the antibody product in different scenarios to ensure that they are sensitive enough for different applications. And here I will show you some of the tests that we perform to compare the antibody sensitivity of our Re-Affinity clone. So of course the performance is tested of the respective application. So I know I'm talking mainly about the flow application, but as I mentioned, we also test their performance in microscopy or mass cytometry when applicable. Further on, we perform the performance of the clone compared to the alternative hybridoma clone, and to test their sensitivity, and we also display this data. I will show you in one slide later. We also test the performance upon fixation, and also performance in case of multi-parameter flow cytometry panel. So quickly on the topic of the performance comparison in flow analysis. Here the aim is to demonstrate whether the performance of our Re-Affinity clone is superior or comparable with the intended application or not. Here basically we follow a method of simple comparison of available clone in the market and show how our Re-Affinity is performing compared to the other clones in a compatible fluorochrome conjugate. And this kind of data set will allow you to also perform less validation from your end, as well as give you the confidence about the antibody quality. And such a data you can also access in our product page. The fixation actually is a very important topic, as I can imagine a lot of you perform experiments on cells that are already fixed, or you have to perform certain fixation step, and you need to make sure that the antibody will perform well in such a fixed cells. Here we perform a staining of our antibodies in before and after fixation with formaldehyde. And in a typical case scenario, when we have the data available, you can easily find them in the product page, and you can see the performance of this particular clone here in CD3 REA613 before and after fixation of cells. And this again offer you the less validation effort, as well as support you with decision that whether this is the right antibody for you to test or not. Finally, the performance of the multi-parameter flow cytometry. This is something new that we are trying to investigate, because a lot of these kind of antibodies, of course, in the end are used in a multi-parameter flow panel. And here, of course, we test multiple antibodies together in a panel, and we are trying to build this collection of panels that we call as Miltenyi Biotec tested panel. And we basically show the performance of the panel. Here is a B cell panel data I'm showing you. And we also offer the staining protocol and the detailed composition of the antibody panel, why certain marker was used and contributed to which fluoride and so on. And these kind of data, of course, also not only reduces your validation effort, it also supports you with your basic panel concept if you are trying to start a new panel building and so on. So together with all this validation data, we are trying to offer you the extended validation of the selected clone that are very important and necessary for you to make sure that the performance is right. And as I mentioned, all this data that I have shown you before actually exists in our product page website with a stamp that called as extended validation, and specifically the knockout data, performance comparison, and the fixation data, as well as the epitope computation. Finally, additional documentation support we also offer in the product page, where we show the tested application and customer review, reference, and reagent repository ID, and so on. Now, in the very final few minutes, I would like to discuss some of the final consideration for this whole topic of reproducibility crisis, and what is still need to be done from us and also you as a scientist. With the awareness of the reproducibility crisis, more and more scientific organizations are requiring thorough documentation of research reagents, such as NIH for their grant application, as well as the big journals, Nature, Cell. They request you to submit really thorough documentation about the protocol of research reagent. And of course, as I already discussed throughout my talk, Miltenyi Biotec is trying its best to create transparency among customers about our product by providing detailed information, additional certificates, extended efforts of pre-tested panel, and moreover, a support to our customer with all necessary information through our experienced customer service and support. In addition, we also ensure to actively take part in supporting scientific community with this kind of quest for antibody validation. And here are some of the examples of our past participation in the antibody validation meeting and the initiative from GBSI, as well as, of course, we're constantly publishing a lot of paper where our antibodies are used. Now, one of the final slides, I would also like to highlight what you as a scientist can do to strongly contribute with this reproducibility crisis by testing and reporting the antibody performance in your experiment, and including the detailed protocol in the respective journal, use RRID and search engines like Antibodypedia and CiteApp. And finally, use also high-quality recombinant antibody for your specific application. And here with my last slide, I would like to highlight that with highly validated and recombinantly generated antibodies, you do not need to throw away your precious data. Rather, you can build confident and reproducible results. And with this, I would like to thank you for your attention, and I would be very happy to take any further question you might have.
Thank you, Dr. Roy, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is it possible to use this technique with saliva?
Thanks for your question. I hope you hear me. So you mean saliva? So our antibodies, of course, are targeted for markers that are expressed in certain cell population, mainly. So if there are certain cell population that you are interested in within saliva sample, I mean, we haven't really tested with such a specific sample type, but I believe if there are cell types that exist for which we have markers, it may work. Yeah.
Thank you, Dr. Roy. Our next question is, how many Miltoni tested panels are out there?
So it's probably referring to the tested panel collection that I was showing previously. So we just, like I mentioned during the presentation, we just started with this project. Our aim is basically to test as more and more panel that are possible, that covers different types of cells, immune cells, and also other cell types from primary origin. So far, we have tested round about 34 panels, but we aim to test more and include more species, and yeah, also, like I said, different sample origin from different tissue and so on. So yeah.
Thank you, Dr. Roy. Our next question looks like a two-part question. Is it correct that only one isotype control is enough for Re-Affinity antibodies? And how about the fluorochrome controls?
Yeah, that's a good question. We also sometimes get this question very often from our customer. As I showed you, that the antibody was built on human IgG1 isotype. So if your panel consists of markers that are just in one fluorochrome, which of course is not always the case. In that case, of course, you use the same isotype. If you have multiple fluorochrome, obviously, because the conjugate will perform differently, in that case, one has to include the multiple fluorochrome. But this reduces the inventory management in the sense that in your bucket of all antibody panel, all you need to have just a few different fluorochrome conjugated to this REA backbone that we call this human IgG1. So if you have that, that should cover all the panels that you usually test.
Thank you, Dr. Roy. Yeah. Our next question is, does Fc receptor mutation in Re-Affinity antibodies abolish all background noise?
So it's specifically to the Fc gamma receptor. So the mutation that we have introduced within this Fc region, it's particularly targeted with this unspecific binding to the Fc gamma receptor, the versions that I have shown you in one of the data example. The aim is basically to kind of block this kind of binding that are originating for this Fc gamma receptor 1, 2A, 2B, and those superfamily, because these are coming from mainly from the larger cell types, monocyte. So sometimes this background gives you like false positive. You may have background that are originated from dirty samples, and like sample that you were working with your gut, from the gut origin or origin which contains some debris and stuff. That I think it's not going to work for these kind of samples. So when it comes to specifically this kind of background that you expect to come from Fc gamma origin, there obviously this will work. I hope that answers. Yeah.
Thank you, Dr. Roy. And our next question here This person says, to my understanding, this is not because one did not detect an antigen that is not present. How do you make sure there the protein is not present? Do you use an alternative technique like mass spec or other antibodies?
Let me get it one more time. So one did not antigen that it is not present. How do you make sure there are that the protein is not present? So if I understand correctly, you were referring to the knockout data that we showed. So in specific for the knockout generation, I mean, we have a R&D team that is really screening a lot of cell line to generate this kind of knockout cells, and they basically screen the cell which expresses this protein, and then take those cells and target the specific region which is responsible for this protein expression, and specifically mutate that via CRISPR-Cas system. I have to check back if they perform any specific test to confirm if this is completely knocked out or not, but I remember, at least when the R&D team explained that to me, that this is the case. So, and of course, then we show this via microscopy data as well as this flow cytometry test to confirm that the protein is completely abolished when checked via antibody binding. Yeah.
Oh, thank you, Dr. Roy.
But if you have further questions, please get back, get into touch with us. I mean, if you have a very specific question, I mean, I'm sure we can get some information from our internal R&D team. Yeah.
Great, thank you. I'm sure people appreciate that, being able to get a little bit more specific, and probably easier to do that on email sometimes. Okay, so the next question. Do you have extended validation data for all Re-Affinity antibodies?
That's a fair question. I mean, of course, as you can imagine, all these tests takes a huge amount of effort. We are obviously trying our best to keep up with as much as possible to add more data for all the antibodies, but at the moment we don't have for all our antibodies, but I believe we have already more than 300 clones for which we have done this extended validation, and this project is continuing, and we definitely would like to continue this and prove the validity of our antibody as much as possible with this kind of data set. Yes.
Thank you, Dr. Roy. So we have another question similar to the last question from the same person. So the same question applies to but applied to specificity, competition assay. So he says, this is not because the signal is low, then the epitope is not present. The antibody may not have access to the epitope for some other reason. Did you try targeted mutations in the epitope? ALA scan, for instance, to assess specificity.
That's a good point. I mean, honestly, with the easy answer, no, we haven't done such an extensive level to targeting, do some targeted mutations. As I explained, I mean, you are right. I mean, there can be definitely other effect, but in principle, we have actually followed some of the tests that have been explained in the field for testing these kind of epitope binding in that regard. So we just followed these assays. But this is the feedback I can definitely bring back with me and discuss with our team and see if there is any additional assay they could perform to confirm that there is no any other additional effect on the competition assay in that sense. Thanks for the question, though.
Thank you, Dr. Roy. And we've got one last question. Finally, are you able to produce in GMP environment?
Yeah, so currently with GMP environment, I mean, we still have some use of some serum in the production system, so it cannot be completely produced in a GMP environment, but the R&D is trying with that to bring the recombinant technology within the GMP environment. And yeah, I can only say hopefully we'll see some of our antibodies and be able to offer them in a GMP condition in the future. Yeah.
Thank you, Dr. Roy. Do you have any final comments for our audience today?
Yeah. So thanks again for taking part and all your questions. I mean, I hope I could bring back the extensive effort that within the team that are trying to perform to validate our antibodies and so on. So if you have any further feedback and you are interested to learn more or want to get any additional information, please feel free to reach out via email. I would be very happy to give you further feedback if possible. That's all.
Thank you again, Dr. Roy, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.